Hi everyone, my name is Arutha Kulasinghe. I'm at the Frazer Institute at the University of Queensland, and today I'll be talking to you about a poster we've done with two brilliant grad students, Habib and Vahid, looking at the ISPLA assay with Navenci. And it was looking at a cohort of 18 head and neck cancer tumors, where we were looking at high dimensional, high plex 60 biomarker information on the tumor microenvironment, and we were pairing it with a serial section PLA assay, a proximity ligation assay, to look at the activation zones of where you've actually got PD-1 and PD-L1 interacting. And so when you have this happening, you have rolling circle amplification and fluorescent signal that you can visualize directly on the tumor microenvironment. And so this is some of the workflows. So we use a PhenoCycler fusion platform, and then ran a serial section to that sample to look at the proximity ligation using the ISPLA from Navenci. And so some of the cohort information for the 18 patients is shown here, and some of the numbers of samples where we got PLA positivity is shown down here. And so we could run the PCF assay followed by the ISPLA to understand what was going on. And the beauty of the study is we have clinical endpoints for therapy. So whether the patients had a complete response, progressive disease, we had all that phenotype information. And so we could look at whole tissue structures, so the whole tumor microenvironment, looking at CD20, CD4, CD8, CD68, pan-cytokeratin, PD-1, PD-L1 expression, so protein expression, as well as activation zones within the microenvironment. And so what we find are that using the PLA assay, the ISPLA assay from Navenci, is that we're finding these immune-rich rivers within the microenvironment which are lighting up 4 levels of activity. And so we can quantify this information once we've done the cell typing, and we've built out the neighborhood maps to understand the microenvironment at a greater depth. So when we look at the areas of activity using the ISPLA assay across these whole sections, we find that in patients that have pathological complete responses to immunotherapy in this head and neck cohort have increased levels of PLA activity zones, whereas the patients that have a progressive disease phenotype have lower levels of activity. And so this makes sense to us because you are looking at that engagement and that activity within the microenvironment. We've also generated some cell type information here, looking at the composition, so you can see where the DC cells are, proliferating tumor cells, CD8s, CD4s, and so on, and the neighborhood masks, which map back to the tumor microenvironment.